I'm Alex. I'm a fourth-year grad student at UC Santa Cruz, and I'm very grateful to have the opportunity today to tell you a little bit about the research that I have been working on lately. So my research focuses on applying computational methods to pathogen genomics at the scale of pandemics. So as you're probably aware, SARS-CoV-2 has generated a huge genomic data set. There's been over 16 million SARS-CoV-2 genomes sequenced and assembled that are now available online. And with this huge data set, there are a number of computational challenges that arise just due to the sheer volume. So what my lab focuses on is how to apply high-performance computational techniques in order to process this huge amount of data. And one of the main things that we look at are phylogenetic trees and how we can build phylogenetic trees using huge amounts of data. And briefly, a phylogenetic tree is just a way to describe evolutionary relationships between genomes. So if you have a sample genome of coronavirus, for example, these are represented at the leaves of the tree, and the internal nodes of the tree represent inferred ancestors of the sample genomes that you've observed. And inferring phylogenetic trees is a very important goal in pathogen research and response because we can do things like lineage designation and assignment, like in SARS-CoV-2, the Omicron, Delta, and various lineages. We can track the global spread of viruses. We can infer recombination events, and a lot of other useful things from this phylogenetic data. So there are two main innovations I want to bring up that have emerged from the SARS-CoV-2 pandemic. The first is the creation of a continuously growing global tree that represents the entirety of global SARS-CoV-2 data. And the second is a tool that lets you visualize million sample trees and the genomes within them at the same time. So USHER stands for ultra-fast sample placement on existing trees, is a tool for creating huge phylogenetic trees using millions of genomes at a time. And USHER has been applied to SARS-CoV-2 throughout the pandemic, and we now have a global reference tree that contains over 16 million SARS-CoV-2 genomes and the evolutionary relationships within them. So the second tool that I want to talk about is a tool called the Genome Browser, which is a tool for visualizing millions of genomes in the context of their phylogenetic relationship. So this tree here is the full global SARS-CoV-2 tree, and each of the colored lines on the right represents a mutation from the reference aligned with the node that it occurs in. And we can zoom in to individual samples or look at the whole tree at once. And we can visualize mutations within specific regions of the SARS-CoV-2 genome, as well as load genome annotations that we pull automatically from the UCSC Genome Browser. And with that, I'd like to say a huge thank you to the ARCS Foundation, first of all, for this amazing opportunity, as well as to all the sources of funding and my awesome collaborators that have made this work possible. Thanks.